Hello, in this video we're going to be talking about genes, general information for genes. So let's not waste any of your time, let's get right to it. Now when we're talking about genes, the first thing we want to think about is chromosomes. Okay, chromosomes are the long strands of DNA, but they're in sections, and each of those sections are called a gene, and those genes express themselves as one, well, I shouldn't say one, as a particular trait. You can think height, you can think weight, you can think, body shape, hair color, hair texture, hair width as far as the individual strands, not the length of your hair, obviously. But that's what a gene does. It's what a gene expresses. And one of the things that we're going to look at in this video is how scientists, and we're actually going to look at it in a couple of videos, how scientists, in trying to understand genes and what they do, isolate them and to see if they can determine what trait they're expressing. And of course that's a big part of what the Human Genome Project was in the 90s and 2000s. The idea of not only what every chromosome does, but what every section of each chromosome, each gene does, and trying to isolate that. And when they figure that out, when they figure out what a particular gene does, what trait it expresses, then you can go to work on applications, commercial applications, as far as products and understanding. I mean, if you figured out what gene causes or is connected to high blood pressure, I mean maybe the gene that helps to develop the blood vessels, if you can isolate that gene, and then maybe you can do some work to help promote that or whatever. But anyway, that's the application side of all that. Beadle and Tatum, as you can see here, it's a diagram beside me there. They studied a fungus, a bread mold, Nersole coraza, but certainly want to ask you that. And they used that fungus to study not only its individual chromosomes, as I've mentioned, but the individual genes on the chromosomes. And they used X-ray to kind of damage and mutate the gene. And what the purpose of is then if they could see what didn't develop later on, if they could isolate a particular gene that they mutated, and if they knew what then they could either see, well, it's one or two ways. They either knew what it was going to produce and see how it changed, or they could see what wasn't produced, later on, and to connect it with that. So as you can see here, what they essentially did is they took the fungus that we talked about, and using X-rays to mutinize or essentially just damage the fungus, and then they just grew it, and to see what things were different. Maybe what in this particular case they were after amino acids were not either produced or were not added to RNA strands. And what their big goal was is to develop a one gene, one amino acid kind of a relationship. And they concluded that there are genes that are associated with just individual or particular amino acids. Now, the next thing we want to talk about is the idea of kind of the principles of genetic code. This is kind of similar to like with Newton's laws. You've got the law of gravitation. You've got the law of thermodynamics. You've got these laws, and they essentially just tell you how things work or what is observed in those scenarios. And the genetic code is no different. It's called the central dogma, and that's simply a reference to the fact that the genetic information flows from DNA into RNA, and then and expressed as a protein. And Francis Crick and his team was one that developed this, but that's just the direction it flows. It never goes the opposite way. You don't take proteins and make RNA. You don't take RNA and make DNA. And this is a two-step process. Transcription, if you know, this word kind of comes from court reporters transcribing, or it doesn't have to be a court reporter, anybody that just transcribes what people are saying into written dialogue, or typed dialogue, I should say. You got a court reporter, and they're simply typing down everything that everybody says, and then that can be put into a document as kind of like not a screenplay, but just an account of every word that was spoken in a court trial. So it's gone from verbal, transcribed, into typed. And so transcription is just the idea of DNA being transcribed into RNA. And hopefully that's not an unknown topic, that you've got some familiarity with that in your high school days. And then the second step is translation. Just like if you translate English into Spanish or Spanish into Japanese, you're taking one thing and converting it into something else. And so then the RNA is translated into proteins, and that's going to be the major topic of this lecture series, how that process happens both in eukaryotic cells and in prokaryotic cells. And typically there's a lot of differences between those two cells. In this case, you're going to see there are some differences, but there's a lot more similarities, simply because we're talking about DNA, we're talking about converting it to RNA, we're talking about ribosomes, and those things kind of all do the same thing regardless of where they are. So, but we're going to see that. But just remember that you're going to see some references to monomers of DNA and RNA in this lecture series. Just remember that's the nucleotides. Those A, T, C, and G for DNA, and an A, U, C, and G for RNA, which means that there are a total of 5 different nucleotides that are used between the two. A, T, C, and G for DNA, and A, U, C, G for RNA. Well, that brings us to the end of our first video in the idea of genes. When we come back next time, we're going to be looking at the genetic code. We'll see you then.